So we're going to explore proteins now, and proteins are biomolecules that comprise 20 to 30% of the cell's mass. They have the most varied functions of any molecules. They include structural, chemical like enzymes, which help with reactions and contraction. They contain the elements carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur and phosphorus. They are polymers of amino acid, monomers held together by peptide bonds. So that's the term to be aware of. The bonds that hold, the covalent bonds that hold amino acids together are called peptide bonds. And the shape and function are going to be due to 4 structural levels. We're going to look at those structural levels to see how proteins are put together and ultimately explaining their function. So all proteins are made up of 20 types of amino acids. The amino acids are joined by covalent bonds called peptide bonds. The amino acid contains an amine group and an acid group. It's actually a carboxyl group that has an acidic nature to it, which means it can give up protons to solution. So they can act as either an acid or a base. The 20 amino acids differ by their R group. So if we look, there's going to be a central carbon. We're going to have a hydrogen attached to it. And on one side, we're going to have a carboxyl group. It's this hydrogen right here that can break off and give a proton to solution. The other side is the amino group, which is a nitrogen and 2 hydrogens. And what makes all 20 amino acids different is right here on the side chain, which we're going to call R. Some R groups are non-polar, some are polar, and some are ionic. So this gives the amino acids different properties overall. So there's your basic structure. Very similar to the drawing I drew, except the R is rotated to the top and the hydrogen is on the bottom. So that's that side chain that we were talking about, or that I was mentioning, that differs in its properties. So bonding between amino acids is going to be the same way. We're going to have a dehydration synthesis. The reaction would occur here between the carboxyl of one group, where we have an OH, and the amino of the other. So that OH and the H can combine to form water. And then we're going to get a covalent bond, specifically called a peptide bond, between 2 amino acids. Here we'd call this a dipeptide for the 2 amino acids. And we have many together, then we would call the chain of amino acids polypeptide. Now, if we want to break that peptide covalent bond, we would have to add water, this way we would, and that would be basically a hydrolysis reaction, same as with the other molecules. There's 4 levels of protein structure which determine shape and function. There's the primary, which is basically just the sequence of amino acids, which, by the way, the sequence of amino acids in a polypeptide chain is determined by genes found in DNA. Okay. The secondary structure is going to be the primary structure as it begins to fold and twist, and this is due to interactions with nearby amino acids. And there's 2 general types. We have alpha helix, which is a coiling and resembles a spring. And then we have beta pleated sheets, which resemble accordion ribbons. And then we have a tertiary structure. Here we're going to have further folding upon itself, the polypeptide. And here, this is going to be due to interactions with the side chains from the amino acids. And then sometimes we have proteins that are made of more than one polypeptide chain. So we have 2 or more different polypeptides together, and that's called the quaternary structure. So not all proteins, functional proteins, are quaternary, but there are many that are. So this is just illustrating that. There's your primary structure, which is just the sequence of amino acids and the order in which these amino acids are put together. Remember, there's 20 different kinds of amino acids, and a polypeptide chain can be hundreds to thousands of amino acids long. The order of those amino acids is determined by the code, genetic code, or the coding within your genes of your DNA. Then the secondary structure, here's your alpha helix right here. And the alpha helix there shows a coiling there, and that's due to interactions between adjacent amino acids. And then the beta pleated sheets also, again, interactions, and you can see these interactions include hydrogen bonding between those adjacent or nearby amino acids. And then if we have further folding of secondary structure, then we get tertiary structure. And here, this is usually due to interactions with the side chains, the R groups within there, causing this folding here. They give an example of a protein called transthyretin, which helps transport thyroid hormone in your blood. And as it turns out, transthyretin is quaternary in structure, so it requires more than one polypeptide chain. So here we have 2 polypeptide chains there for an actual functional transthyretin molecule. Shapes of proteins fall into one of 2 categories. There can be fibrous or globular. So the fibrous are referred to sometimes as structural proteins. They're strand-like and water insoluble, which means they don't dissolve in water, and they are relatively stable. Most are going to have tertiary quaternary structure, which we just discussed previously, and their role is to provide mechanical support and tensile strength. And examples would be like keratin, which makes up your fingernails and your hair, elastin, which is a more flexible fibrous protein, collagen, which is the most abundant protein in the body, and you find that in your skin, you find it in your bones, and so on. And then contractile proteins like actin, which is found in muscle cells to help shorten the muscle cell. And then we have globular or functional proteins. So these globular proteins, they're going to be compact, spherical, and they're going to be dissolved in water. And they are going to be sensitive to environmental changes. So if the environment changes significantly, and here's why it's important to maintain homeostasis, if homeostasis is disrupted for a number of factors, including temperature and pH, that can change the shape and of the protein, of the globular protein, and if that shape changes, then the function may change. So they are tertiary or quaternary in structure. They have specific functional regions or active sites. Examples of these globular proteins include antibodies, which help protect us when we've been exposed to some kind of disease agent, hormones like insulin, molecular chaperones, which help fold proteins, and then enzymes, which are the biological catalysts to help your metabolism run. Globular proteins can become denatured. We call that denaturation. And this is where the proteins unfold and lose their functional 3D shape. Okay. Fibrous proteins are not as able to denature, so they're more stable. But as far as globular proteins, they can become deactivated because their shape changes. So their active sites literally change shape and then can no longer function the way they're supposed to. The denaturation can be caused by changes in pH and increases in temperature. These the shape change can be reversible if normal conditions are restored. However, it'll be irreversible if the changes are extreme. Example here would be like cooking an egg. When you cook an egg, the proteins that make up the egg white change, and you can't change them back. Enzymes. Enzymes are specific globular proteins that act as catalysts. And catalysts, basically in general, are going to be substances that assist and speed up chemical reactions. And within your system, these catalysts, biological catalysts, are enzymes, and they help run your metabolism. The reactions in your cells would not be possible without the help of these enzymes. So they are catalysts that regulate and increase speed of chemical reactions without getting used up in the process. So that's the key. The catalyst remains there; it just assists reactants to go through their reactions. And they do this by lowering the energy needed to initiate the chemical reaction. So they lower this, what's called activation energy for the chemical reaction. What does this do? This leads to an increase in the speed of the reaction, and it allows millions of reactions per minute to occur within your cells. So what are the characteristics of enzymes? Most functional enzymes are referred to as hollow enzymes, and they consist of two parts: the apoenzyme, which is the protein portion, and some cofactor like a metal ion, or a coenzyme, which is some organic molecule like a vitamin. Okay? And without those cofactor coenzymes, the hollow enzyme would not be functional. Enzymes are very specific. That's a characteristic. That means they act on a very specific substrate. So the enzyme, for example, that breaks down starch in your saliva is called amylase. It's specific to work on starch. You can usually recognize enzymes because they end in the suffix ase, like hydrolases, oxidases, amylase that I mentioned a while ago, alcohol dehydrogenase, acetylcholinesterase. All of those you already recognize they're an enzyme because of the suffix ase. They end in ace. So how does the enzyme action work? The enzyme is going to lower, I mentioned this earlier, activation energy, which is the energy needed to initiate a chemical reaction. So what enzymes do actually, they prime the reaction, allowing it to happen. Enzymes allow chemical reactions to proceed quickly at our normal body temperatures. There's 3 steps in this process involved in enzyme action. The substrate, which is what binds to the enzyme, the active site, is going to temporarily form an enzyme substrate complex. So we have to have substrate binding forming enzyme substrate complex. The complex is going to undergo a rearrangement of the substrate, resulting in a final product. The product is then released from the enzyme, and the enzyme is free to work on the next substrate. So here's an illustration of what happens here. Normally, let's say we have 2 reactants and they have a certain energy level. This on this y-axis here on this graph, that represents energy. And in order to get the reactants to react, we need to actually give it a push. So energy is required. And so we have to get over this hill right here. And if we can't get over that hill, then the reaction will not happen. Sometimes we get to that top of the hill by increasing temperature. Only thing is we cannot increase temperature too much, or that'll mess up the enzyme. So and other important parts of running our metabolism. So instead, we have enzymes, and the enzymes are going to grab onto these substrates, and what they do is they end up lowering that hill. So they work by lowering our activation energy. So when the enzyme grabs onto the reactants, it facilitates the reaction, or it makes it easier to happen. Now notice the same change in energy. In both cases on the left, without the enzyme, we start at this energy level where that dashed line is. It's the same in both graphs. And we end at the same level here. We have the same final energy. So ultimately, the same energy transformation occurs. Overall, the same net change. When we go up in energy, if we add energy and then we come right back down to that line, it's like there's no net change in energy. The change is 0. It's like throwing a ball up. Ball goes up, gains potential energy, and loses kinetic energy, and then comes back. It gains the energy back that it lost on the way up. So there's no net change in energy. But if that ball were to allow to continue to fall, because I drop it instead of catching it higher up, now there's going to be a net change in the energy. Same thing here. And this net change is the same regardless. So we start here at E1, and then we end at energy level 2 down here. And that change is the same in both graphs. We start at E1, it's the same, and we end at E2. If we subtract the overall energy there, the change in energy is the same. So it doesn't change the nature of the energy associated with the reactants or the products. What it does is lowers the activation energy. So here's our model that we were looking at or explaining in a previous slide here. So here we have substrates. Let's say this is an enzyme that's involved in helping to make a peptide bond between 2 amino acids. And so our enzyme has an active site, and that active site is very specific for our substrate. It's not going to bind to anything else except the amino acids or our substrate, whatever our substrate is. So the substrate finds the enzyme, and when bound to the enzyme, forming the enzyme substrate complex here, that enzyme substrate complex is going to change the nature of the energy of the reactants, the substrate, and make it easier for the reaction to occur. And so here, If we're going to form this dipeptide here in the substrate here, we're going to have to remove a water, but the enzyme is going to help that process occur. Once the enzyme has finished helping to cause the reaction, it's going to release the product, and the enzyme is free to bond again with more substrate, in this case the amino acids from the beginning.